Kenta's Adventure of Biotechnology. Kenta young farmer. Recent challenges. Low harvest yields. Crop diseases or health-related issues.
Oh dear Kenta, feeling a bit puzzled? Wondering how to grow better crops or fix what's gone wrong? Let me share a secret with you. You have a special friend called biotechnology. There are two distinct approaches, and I'll show you what each one needs, along with the tiny wonders that make them work.
Classical processes. Genetic engineering.
Hello and thank you. The vegetables in my field, especially the tomatoes, are struggling with disease and producing very small fruits. Do you think traditional methods could help improve their health and yield?
Sure. Here are 3 methods you can try.
Selective breeding. Choose the strongest, healthiest plants and grow them year after year. Little by little, they improve. Cross-breeding. Combine 2 different plants to mix their best traits. This is called heterosis, hybrid vigor. Mutation breeding. Use radiation or chemicals to create changes in the genes, and uncover exciting new traits.
Selective breeding. Selection is based on visible or measurable traits, such as crop yield, fruit size, and resistance to pests and diseases. Sometimes, DNA markers are also used to guide the process. Here are some examples of plants developed through selective breeding. Cross-breeding. This method involves crossing plant varieties with different characteristics, then selecting offspring that show the desired traits. Over time, this process helps improve the variety. For example, by crossing a disease-resistant variety with one known for great taste, it's possible to develop a new variety that is both resilient and delicious. Mutation breeding. Sometimes nature just needs a little nudge. By using radiation or chemicals, we can create small changes in a plant's DNA. Occasionally, these changes lead to useful new traits, and we select the best ones.
Oh, that sounds great. It's really interesting. Should I give it a try right now?
Yes, those methods are known to be very good in terms of harmony with nature and safety. However, each method does come with a few things to watch out for.
Selective breeding. It can take a long time for desired traits to emerge, and genetic diversity may decrease over generations. Cross-breeding. Unexpected gene combinations can occur, sometimes bringing unwanted traits along with the desired ones. Mutation breeding. Since many mutations are random, finding useful ones requires effort, and it takes time to stabilize a new variety.
In addition, we are confronting the serious and rapidly accelerating challenge of global warming, which requires new and innovative approaches, as current methodologies are too slow to keep pace with changes such as increased rainfall. Recently, emerging technologies like genetic modification and genome editing have shown promise in addressing this issue. To understand them, it's important to first learn the basic terms. It might seem challenging, so I'll start with the simple concepts. Let's begin with DNA. To conduct experiments, you need a sufficient amount of DNA. To solve this, scientists developed a clever method to artificially synthesize DNA, known as PCR. Here's how it works. Have you heard of plasmids? They're small, circular pieces of DNA found in bacteria that can replicate independently. Scientists use them as the vectors to deliver specific genes into cells.
Insert the desired gene into a plasmid. Introduce the recombinant plasmid into bacterial cells. Let the bacteria express the gene and produce the target protein.
That was some basic information about genetic modification. Now, let's move on to genome editing. Unlike genetic modification, which involves inserting genes from other organisms, genome editing can also use the organism's natural repair mechanisms to modify its existing DNA, allowing for changes identical to those produced by traditional breeding methods. The methods I've explained are under active research and undergo strict safety testing to ensure that their benefits outweigh the risks. Some of these technologies are not yet in practical use due to regulatory restrictions, but biotechnology holds great promise for supporting human survival while also protecting the environment.
Technology has advanced so much. Although some parts were a bit difficult, learning about biotechnology really sparked my interest. It seems to have applications not only in the crops I grow, but also in treating diseases in humans and animals. I want to learn more, especially after discovering that genetically modified organisms, GMOs, can even be considered organic.
In the heart of the forest, where the green leaves glow, tiny wings flutter where the healing winds blow. We dance with the data, we sing with the trees. Biotech fairies on a mission of peace. On DNA spirals in the sky, nature and science flying high. With every gene we plant a dream of cleaner air and crystal streams. We're the fairies of the code of life, spreading hope, not harm or strife. We whisper to flowers, we talk to the bees. We mend broken roots and revive dying seas. With glowing test tubes and chlorophyll light, we bring back the balance, we make it right. From glowing mushrooms to healing vines, we blend the magic with biotech signs. CRISPR wands and petal shields, guarding forests, farms and fields. On a DNA spirals in the sky, nature and science flying high. With every gene we plant a dream of cleaner air and crystal streams. We're the fairies of the code of life, spreading hope, not harm or strife. From glowing mushrooms to healing vines, we blend the magic with biotech signs. CRISPR wands and petal shields, guarding forests, farms and fields. On a DNA spirals in the sky, nature and science flying high. With every gene we plant a dream of cleaner air and crystal streams. We're the fairies of the code of life, spreading hope, not harm or strife.